This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM This Week in Microbiology, episode 196, recorded on March 28th, 2019. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello. How are you?
Good to hear you. I haven't heard you in a while.
It's been a while, indeed. So I'm glad good to be back.
It is good to be back. In fact, we have the whole crew here today. Joining us from Ann Arbor, Michigan, Michele Swanson.
Hello, where it is a 17-degree Celsius day.
It's cold. Wow.
63 degrees Fahrenheit. I'm sorry, I goofed that up. It's 63 degrees Fahrenheit.
That's right. It's 17, yeah. Well, winter's, I think, gone pretty much. Although I remember one year it did snow on Easter. That's many years ago. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. It's also 17 degrees or 63 degrees Fahrenheit here in beautiful sunny Charleston here today.
Charleston.
All right.
We're all together. We haven't been together in a while, so it's really nice. In fact, people often comment on Twitter, where was so-and-so, where was so-and-so? And here we are. We're all here today. Hope you enjoy it. We have a snippet and a paper for you today. And we're going to start with Elio, who's got a snippet for us.
Okay, the snippet and the paper are as unrelated as can get.
You bet.
There's no doubt.
Very cool. Very cool.
Mine
choices.
is about farming and alcohol. But I don't mean people in Minnesota doing that. I mean beetles. Okay, let me start over. You would think that farming was invented in the Mesopotamian, in the Middle East, sometime in some 30, 40
a long while
years ago, right? Not so. It was invented 40 to 100 million years ago by ants, termites, and beetles. They all have the ability to cultivate, and what they cultivate is fungi. Now, many of the listeners will have heard of leaf-cutting ants. So let me start with that for a little bit. Leaf-cutting ants are ants in the tropics, the attines, which go out and harvest bits of leaf, and they bring it back to the nest. And the sophisticated listeners know that it's not the leaves that they eat. Not that. They chew on that, they masticate them, they make it into a pulp, and they seed them with fungi. They grow the fungi and they prune them to avoid them becoming mushrooms, which they couldn't use. So they keep them as the equivalent of molds, which grow on this masticated, chewed up leaves. And the fungi are their sole sustenance. That's all they eat. Without fungi, they would die. Okay. Now, when they make a new colony, the queen makes sure that she carries this fungi with her, and she does that by having a special organ called the infrabuccal organ. It's right underneath the mouth. There's a space in which she packs it full of hyphae, fungi, and goes off to start a new colony. And the first thing she does is she finds some leaves that are chewed up and inoculates them with the fungi, thus establishing continuity. Termites do something similar. Now let me go on to the beetles. Gardeners know about Ambrosia beetles. They're a pest. But they're a relative pest, because they tend to infect trees, not live trees so much as diseased trees or dying trees. It depends. There's some Ambrosia beetles in San Diego County which affect live trees, but generally they don't. So what they do is the females tunnel into the wood, and they seed that with fungi, which they brought in an organ, not the infrabuccal organ, but a thing called a mycangium. A mycangium is an organ that's packed full of spores of fungi, and that's on the back. Anyhow, whatever, they extract some of the sap, essentially the sap, which they somehow it's mixed with some of the feces and so forth. It becomes something which they call ambrosia. Ambrosia, I remind you, is the sweet nectar-like food of the gods. In this case, it's not the food of the gods, it's the food of the beetles, the Ambrosia beetles. So they put this material on the walls of the tunnels, and they seed it with the fungi which they brought along in their mycangium. Now, here's the story. The fungi that grow there are very special. In other words, they have a problem because this is rich, the substrate is rich, biological material, rich in foodstuff, and it would be a perfect growth medium for contaminating fungi, which the ambrosia cannot eat. Now, the ants have exactly the same problem because their chewed up leaves would be a great place for Aspergillus, Penicillium, regular molds from the environment to grow on. So the ants solve the problem neatly by having another bacterium on them, usually Streptomyces, which make antibiotics which are specific against the common molds and do not affect the fungus which they eat. Okay, it's a neat story, isn't it?
It's great. It's just beautiful.
So the same thing. So they have a weed killer. Okay?
Literally a weed killer.
It's a real weed killer. So that's great. But the same thing happens in the ambrosia ants. But in this case, the weed killer is not an antibiotic, but it's alcohol. So here's a story with alcohol and the beetles. First of all, in decaying wood, there is a small amount of alcohol is made by the wood, okay, by the decayment. Now, it turns out that the ambrosia beetles like alcohol. They're attracted by it, so that if you, in an experiment they did, which is really typical of what they did here, they take a bit of wood, a trunk, and they put about a certain amount of alcohol on one end of the trunk, and they wait and see what happens. So it turns out that ambrosia beetles are attracted to that, and the concentration of ambrosia beetles in the end of the trunk, which has alcohol, is far greater than that in the other end. Okay.
So the beetle knows that the tree has fallen or is beginning to decay by virtue of the fact that it's making a higher concentration of ethanol than the healthy trees next to it.
That's right. Yeah, I think that's
remarkable.
Right. Yeah, it is remarkable. It's a signal, and the ambrosia beetles like it, and they like about 2% alcohol, which is like a weak beer.
Very weak beer. The kind they serve at baseball stadiums. That's called near beer.
They go towards that. Conveniently, the ethanol in similar concentrations selectively inhibits the unwanted fungi. And there's a whole paper that deals with that, and the paper is by, one second, it'll appear in a second.
Ranger, Biederman, Putenmart, Bagala, Gosh, Palmquist, Muller, Barnett, Schultz, Redding, and Benz.
And they are from all over the place. There are too many institutions, including in Germany, in this country. It's in a whole bunch of universities. So I won't tell you where all this comes from, but it's a multinational and multi-laboratory effort. So this paper, which is entitled Symbiont Selection via Alcohol Benefits Fungus Farming by Ambrosia Beetles. And that's kind of like the story. It's pretty much the story that I have to tell you. It is a little bit more, the paper is quite sophisticated. It shows up, it shows also something very interesting, namely that the ambrosia fungi. Let's call them ambrosia fungi, the ones that grow on the ambrosia and are not inhibited by a small amount of small concentration of alcohol. They happen to have alcohol dehydrogenase activity. So they can get rid of the alcohol, which is essentially deleterious. So that's pretty neat, isn't it?
Yeah.
So whereas the other fungi, the molds, the contaminated molds, do not have it, and therefore they are killed by 2% alcohol.
So they're attracted by it, but then they decompose it because it would kill them.
You mean the contaminating molds, are they attracted to it?
No, the beetles are attracted.
The beetles are attracted. Yeah, the beetles are attracted to it.
But they're eating the fungi that then make the ethanol as a waste product. So they're effectively.
I don't know where it's not clear where the ethanol comes from. It is typical of decaying trees, but I don't know who's responsible for making it. You're right. It may be regular contaminating fungi, molds, like penicillium and aspergillus. I don't think it says that in the paper, so I don't know where the alcohol comes from. But it's a fact that decaying wood has a certain amount of alcohol, just the right amount to inhibit penicillium, aspergillus, and allow the growth of the ambrosia fungi, which are called Ambrosiella and Racaniello.
For the tree biologists that are listening, I just want to add one point. Is the authors point out in their introduction that ethanol is a byproduct of anaerobic respiration, and it's actually ubiquitous in sapwood, phloem, and cambrium of many living tree species. So I guess there's some low level even in a viable, healthy tree, but maybe higher levels than if the tree is suffering. I
suppose it's because there's anaerobic respiration by bacteria in trees.
They also point out in the last part of their discussion that the ethanol is a stress-induced response by the dying tree. So as the tree begins
to... That's true for dying
trees,
but it's also true in healthy trees.
Yes, it does produce a basal level of ethanol due to anaerobic respiration.
Right. So we don't quite know where the ethanol comes from, but it's there both in healthy trees and in dying trees. And the Ambrosia beetles love it, and the ambrosial fungi are not killed by a small concentration of it. So it's a neat story. So here's farming, which includes, what does it include? It includes preparing the soil, fertilizing it, which in this case is the ambrosia, seeding it with the proper fungi, which the beetles do by virtue of having this mycangia, pruning it because they get rid of or inhibiting the growth of weeds, killing weeds, and harvesting it. So what else do farmers do? That's exactly what farmers do.
The only thing they didn't do is take it to market.
Yeah, that's right.
That's right.
They didn't sell
that's right.
it.
However, they take it with them when they travel. So
I guess I'll concede that point to you. They are taking it to market when they move down the road to another tree.
Right.
So they are indeed our first farmers.
Elio, how do they apply the weed killer? How do they take the ethanol and put it on the gardens? I don't get that part.
No, it's right there. I don't think they have to do that. It's just simply there.
I see. So they make gardens only where there is ethanol because they're attracted to it, right?
That's right. They're attracted to ethanol. It's a good place for them. So the beetles build a tunnel. They get the ambrosia. They seed it. They do what farmers do.
And they eat. They live in trees. Is that correct? They bore.
That's right.
They make tunnels in trees.
They are a pest in the sense that if they sometimes, as I say, they affect a non-dying tree, in which case the tree does die. Mm-hmm. So it's possible. So people worry about ambrosia beetles. They're not the friends of the farmer or of the tree grower. But in general, they're not as bad a pest as so many others, wood-boring insects. Mm-hmm. Here they seem to be more at home in dying trees or decaying trees.
And inordinate fondness for beetles. You know that book?
I don't.
Well, it was said by Haldane.
Yeah, it was said by
what
Haldane.
had he learned about the deity from being a biologist all his life?
Yeah.
His answer was just that.
He said he has an inordinate fondness for beetles. And then many years later, E.O. Wilson wrote a book about beetles and called it An Inordinate Fondness for Beetles.
Ah. Well, this story, the biology, was very cool, but I was intrigued by the methods that they were able to bring. So they did a lot of really quantitative dose-response experiments with these living trees and the beetles and the fungi and all quantitative.
Mm-hmm.
So, for example, when they wanted to ask if the beetles truly were attracted to trees that had higher ethanol concentrations, they either cultured a tree without ethanol, or they dosed the soil with different amounts of ethanol and then quantified how many beetles and then how many eggs the beetles laid in the burrows that they created in the tree. So it's really a beautiful quantitative paper creating this lovely story of ecology.
Mm-hmm. It's a wonderful story. I love it. Thank you, Elio.
Of course.
All right, now we move to something very different. We move to your ear. And this paper is all about the ear and bacterial infections. It is a paper published in Cell Host and Microbe. It's called Cleaved Cochlin Sequesters Pseudomonas Aerogenosa and Activates Innate Immunity in the Inner Ear. And the authors are Jung, Yu, Cho, Yoon, Hyun, and Choi from the Yonsei University College of Medicine in Seoul, South Korea, and Hongik University, also in Seoul. And it's very interesting. One of the authors is from the Department of Sculpture at Hongik University.
Yeah, I looked in the acknowledgments. That person contributed technical support. So as you're going to describe, there's a lot of three-dimensional architecture that goes into thinking about this spatial restriction of the inflammation. So I wonder if that person helped with some modeling. Maybe,
yeah. So let me tell you a little bit about the ear. I do not know anything about the ear except that I have two of them. I am by no means, but it's very interesting. And so all of you know, let's take a human ear. Although the studies were done in mice, we have ears that we perceive sound with, of course, and they're amazing. There's the outer ear, right? You have your ear lobes, and then the outer ear kind of funnels the sound in. And you can put your finger in your ear, and that's the outer part. But then there's the tympanum, or the eardrum, which you can get to with a Q-tip, I guess. But that is what senses the vibrations. And then behind the tympanic membrane is the tympanic cavity, or the middle ear. And that is where these wonderful three bones, which I learned years ago, the malleus, the incus, and the stapes. They
never thought you'd need that again, huh?
Oh man, they're so cool. They're such cool little tiny bones, and they're held there by ligaments, and they transmit the vibrations from the tympanic membrane to the cochlea, which is in the inner or internal ear. And cochlea, you may remember, it's that organ that kind of looks like a snail. It's coiled up at one end, and it is full of fluid. As you'll see, it's lymph fluid. And what happens is the sounds that come from the tympanic membrane are led into the cochlea, and the fluid vibrates. It vibrates hairs that are in this organ, and then the hairs are connected to nerves, which go to your brain and sense the sound eventually.
Yeah, hearing is truly a superpower because it enables us to perceive space in 3 dimensions.
Mm-hmm.
Because you only need to have one of your ears blocked or clogged after a plane trip to know that your directionality of sound is gone.
Right.
So it's truly a superpower. That, along with vision, are our 2 human superpowers that allow us to perceive space in 3 dimensions.
Is this as good as Beetles?
I think it's as good as Beetles, but we don't have ethanol yet.
Right.
And here on TWiM, we like ears because that's how you listen to our show. Right?
That's right.
Without ears, you wouldn't listen to them. So the topic of this paper is, as we all know, you get ear infections, right? Starting as a kid, all our kids had ear infections over and over again.
Any parent knows the inflammation that occurs because that is coupled to the screaming and the wailing of your child due to an ear infection.
Yeah, because this is a very delicate organ with fluid in it. And so if bacteria get in it, this cochlea, and you have an inflammatory response, that can actually clear the infection, but it causes swelling and pain. And so there has to be a balance between clearing the infection and not damaging the organ. And this organ, the cochlea, is made up of several ducts. It has structural parts, bony parts, but it also has what are called membranous labyrinths. There is a tympanic duct, there's a cochlear duct, and there's a vestibular duct. And they're kind of parallel ducts that run along this cochlea, and they're full of fluid. If bacteria get in them, that's bad because they can damage you can have long-term hearing damage, and that's not good, obviously. So this paper is trying to understand. We know that these infections can be controlled, but it's trying to understand how the inflammatory reactions that occur are modified so that they don't damage this delicate organ. We don't really understand that. So the organ of Corti in particular has these sensory hairs in it, and how do you prevent damage to that? So,
may I just interject?
Of course.
That organ of Corti is named after an Italian histologist, Alfonso Corti, who was the first to describe this organ back in he was born in 1822, but he published this in 1850, studying at the University of Würzburg using histology to discover this really beautiful anatomy that will become relevant in our story.
It's very hard to study this because the rest of histology is about soft tissues, which you can slice in a microtome very easily. Here you have bone surrounding this, so you have to really have special techniques for making thin sections and doing the anatomy of it. It's very elegant.
And Alfonso Corti did it at a time when he was just using bright field microscopes. So he was putting the tissue slices, under a cover glass and then deducing the structure. So he got a lot of it right, but obviously he'd be very proud to hear us talking about what's known now.
And if you have an opportunity to see the color images associated with this first figure, you can see the beautiful anatomy that he originally described using bright field, but they do it in many colors using sophisticated confocal microscopy.
They use the original Italian names for these, what I call the ducts. They call them the scala vestibuli, the scala media, and the scala timpani. Scala, I guess, means room, right, Elio?
Well, scala means ladder, actually, like La Scala in Milan. But why it's called that, I don't know.
All right, so here's where this paper begins with a protein called cochlin, Cochlin,
which I gotta admit I never heard of.
Exactly, neither have I. But it is abundantly produced in the inner ear and also in the eye and the spleen. And of course, the ear and the eye both have fluid in them, which is important for their function. So that's interesting.
And they're both immune privileged.
They're immune privileged sites. I don't know about what's it doing in the spleen. Maybe it has different functions there. So the protein is interesting because it has a domain called a limulus factor C domain. It has a cochlin domain and an LGL1 or LCCL domain, three separate domains. And this
which you don't have to memorize.
Don't have to memorize. But this LCCL domain has homology to limulus factor C, which is a proteinase involved in immune responses in horseshoe crabs, which of course are limulus. And it's thought to have an antibacterial properties. And mutations in the gene encoding cochlin in people are associated with hearing loss. So there's some evidence that that's important for hearing, cochlin protein. And in the eye, if you have too much cochlin, you get glaucoma. It's associated with glaucoma anyway. So this seems to be an important protein.
Well, it makes sense because if you have too much, the pressure is going to build up in the eye and damage the nerve cells. So intuitively, it makes some sense.
Yep. So in the spleen, cochlin seems to promote systemic innate immunity against bacterial infection. And what they find is that if the spleen is infected, cochlin gets cleaved, and this LCCL domain goes into the blood and can go to other tissues where it promotes innate immune responses. And how it does that is not really understood. So they're thinking, well, cochlin must have an important role in the eye. Maybe it's involved in defending against infection. So they look at Pseudomonas aeruginosa infection in mice and in the ear. And this is one of the bacteria that cause ear infections. And in particular, they look in this fluid that's in the cochlea, which is where bacteria would go and multiply in an ear infection. So again, it has these fluid-filled chambers in the cochlea. And so they do an experiment, which Michael alluded to already, where they stain for the cochlin protein and look by microscopy, and they can see where it's produced. And you can see the protein is very much limited. It's not everywhere, but it's limited to certain places, membranes and ligaments, for example, not flowing through the fluid, right? So it's in very specific places in this multi-chambered structure. They do a really interesting experiment where they take mice and they give them loud, high, certain frequency sound, and then they measure the electrical response in their brain. So they've implanted them with electrodes, and they can basically see when they sense the sound by the brain activity. And so they use higher and higher volumes, basically, and they get a threshold, an auditory threshold, right? And they wanted to know if cochlin had any function in hearing, but they have cochlin knockout mice, and they compare them to wild type, but there's no difference in the threshold of sound that they both perceive. So it's not involved in at least normal auditory function.
Healthy ear.
In the healthy ear.
In the healthy ear.
Yeah.
But just wait. He's going to hit you with the punchline.
So then they put Pseudomonas into the inner ear. So they inoculate the bacterium into the middle ear, and it goes into the inner ear. And then they say, what is the effect of having these bacteria multiplying on auditory function using the same assay? And again, with or without cochlin. And so in wild-type mice, the hearing thresholds are similar. Even in infected mice, they're compared to uninfected mice. But in the cochlin knockout mice, you have to play the trumpet a lot louder for these mice to hear you.
Oh, yeah.
After infection with Pseudomonas. And so that's really cool. I love that.
Well, it explains.
The data are beautiful.
It's visually easy to tell that there's hearing loss in the knockout animal, the Koch minus one. And in the one with the Pseudomonas, it's really pretty remarkable to see what an infection can do. And you just simply look and compare and contrast between the wild type cochlin cultured cells versus those with Pseudomonas, and it's remarkable. I know, but I'm still amazed.
He's amazed by
I can be amazed.
It's okay. I think
I'm allowed to be amazed.
I like the assay because obviously mice can't tell you when they hear something, right? Humans,
they can't raise their hand.
They can't raise their hand. Exactly. I remember that.
Yeah, you raise your hand when you hear.
But so somehow cochlin is involved in preserving auditory or hearing preservation. Let's put it in infections, in Pseudomonas infections. So what's going on? That's what the rest of the paper tries to look at. What is cochlin doing? So first they take explants of the inner ear out of the mice. They take them out and put them in culture. So it's the entire structure, but it's bathed in fluid so that they can infect it. And they can look for the cochlin protein. and they see when they're not infected, their protein is full length. But when they infect these cochlear explants and culture, the cochlin gets cleaved into two fragments. And it looks like one of them is the LCCL domain, this one with homology to the horseshoe crab protein.
Mm-hmm.
And you don't see this in the cochlin knockout mice, so it looks like it's cochlin that's being cleaved. And also, if you put bacteria into the ear of living mice, and you do the same experiment, you see cochlin is also cleaved in vivo. Okay, so
1 second. Isn't this the ability to take the cochlea out and still functioning kind of amazing? You think a nervous system is required, some nerves or something, for the cochlea to function, but it functions perfectly well without it.
Yeah, at least in terms of this
I'm
protein.
amazed by that.
So what is
it an organoid?
Well, I don't know. It's an organ, right?
It's an organ, right?
Because organoids you usually make from stem cells, and they differentiate into what looks like an organ, but it's never complete. That's why we call them organoids.
Uh-huh.
So there's something missing, like immune cells or something. But here they're taking the organ out. It's small enough so that you can do that. We do this sort of with mice. We make brain slices and put them in culture. They're not the whole brain because you can't sustain that. Media doesn't get into it. But if you slice it, the slices are thin enough so that media can bathe it and they can live. And all the cells are there in the brain slices too. Anyway, so what's cleaving cochlin? Well, it looks like they can use a series of inhibitors of proteases. And it looks like one of the enzymes is a matrix metalloproteinase, which are tissue proteinases that have a variety of roles in the mouse. And so that seems to be one. And another protease is called aggrecanase 1 and 2. These seem to be cleaving them as well. So it's being cleaved by cellular proteases during bacterial infection. And where the cleavage product ends up in the lymph in this organ. So remember, the cochlin itself is associated with membranes and ligaments, but after it's cleaved, the LCCL goes into the lymph portion. And that's important for what we're going to see in a moment. By the way, when you infect mice with Pseudomonas, and these mice have no Cochlear gene, if you add exogenous cleaved fragment, LCCL, to them, it lowers their hearing threshold.
Which is good.
Yeah, it's good. Yeah.
In this case.
In this case, it's good. So you're somehow, the LCCL is maybe interfering with bacterial growth or something to allow normal levels of hearing, or at least better than without LCCL. Okay, so what's going on here? The next experiment, they ask, does cleaved cochlin affect the number of bacteria in the inner ear? So they took their cochlear explants from wild type and Koch minus mice. They inoculate them with Pseudomonas, and they measure bacterial growth. And they can see that in the Cochlin minus mice, you get more bacteria, a lot more bacteria than in the explants from wild-type mice. So that suggests that the protein has some kind of antibacterial role. So they want to know how that happens. And they did a series of experiments, which in the end indicate that cochlin is upregulating a number of pro-inflammatory cytokines. These are proteins produced by the innate immune response that are important for clearing infections of various sorts, causing inflammation, the influx of immune cells and things that you need to get rid of infections. And this cochlin seems to be involved in that. And they can add inhibitors of this matrix metalloproteinase and show that the effect of bacterial infection on the induction of cytokines is inhibited when you inhibit the protease. So that means that the cleavage and the secretion of LCCL into this lymph are important for inducing these cytokines that are important for clearance of bacteria. So how does it work? How does LCCL work? So again, they do Western blots to localize where the fragment is, and they only find it in a certain part of the scala Timpani. So you have to, this may be hard for you to visualize, but remember there are three chambers. There's the scala vestibuli. The scala media contains the organ of Corti with the hairs in it that sense the sound. And that's crucial not to let bacteria get in there. Probably ruin the hearing. And then below the scala media is the scala Timpani. So these are all surrounded by membranes, and in the membranes in the tissue around them is where the cochlin is. And when it's cleaved, it's released only into the scala Timpani, not the scala media, not the scala vestibuli. I love saying those words. Okay.
You're doing beautifully.
So cleavage of cochlin produces this soluble fragment that goes into one of these chambers specifically, not just all of them. All right. And so it's the LCCL domain, which has this antibacterial effect. And that seems to limit the bacteria to the scala Timpani. Because if you inoculate mice with the bacteria, in wild-type mice, they're limited to the scala Timpani compartment. But in Koch minus mice, the bacteria are dispersed throughout the cochlea in all these three compartments I just mentioned, including the one where the organ of Corti is, which is not good because it's going
as you can imagine, as the bacteria grow and multiply, the net consequence will be death and destruction to the sensory organ that perceives hearing.
That's right.
And the delicate hair cells on the epithelium that senses sound.
That's right. And that's why if you have multiple ear infections, it can affect your hearing, right? So basically, what they think is happening, the cochlin is in the tissues surrounding these vestibules. When bacteria get in, that stimulates the cleavage, and how that happens, we don't know. The LCCL domain is released. It goes into the scala Timpani, and they think it's sequestering the bacteria there because they don't see it spreading elsewhere. And if you take away the Cochlear gene, then the bacteria can spread elsewhere.
Let's take a closer look and see what's really happening.
Right.
What's the next experiment?
What's the next experiment, Vincent?
So
before we blurt it out.
Next they do some cool immunofluorescence using green fluorescent protein, which allows them to readily see neutrophils and monocytes, which are immune cells that are important for clearance of bacterial infections. And they can see very easily that when you infect mice in the ear again, that these cells come into the scala Timpani after infection of wild-type mice. But in mice lacking the Koch gene, the Cochlear gene, the neutrophils are dispersed throughout it, including in the organ of Corti. And again, that's where the bacteria are. Remember, if you don't have Cochlear gene, the bacteria go everywhere, and so do the neutrophils. And so they're going there because they're trying to clear the infection. They're attracted to whoever the bacteria are, but it's not good because the more cells you have in that small area, it's going to cause damage.
Inflammation, damage to the delicate hair cells, and everything else in there.
So neutrophils, also macrophages are attracted in a cochlear-dependent way, and even dendritic cells. All of them are attracted, and they're limited to, again, the scala Timpani by the presence of LCCL there. They also do a lot of nice stainings of cytokines as well, and they can see that cytokines go where LCC is. And if there's no cochlin, then everything is dispersed throughout the organ, which again is not good, and it can damage everything. So the idea here is that cochlin enhances the induction of inflammatory cells and cytokines, but just in the scala Timpani, and that's where the bacteria are. So why are the bacteria just in the scala Timpani? Well, they think that...
or why are they stuck there?
Why are they stuck there? So they show first that bacterial viability isn't affected by the LCCL peptide. If you try and grow the bacteria in vitro and add the peptide, it doesn't affect their growth. But they noticed that when you add the protein to bacteria, it kind of aggregates them, makes globular aggregates. And they say it's not a biofilm, but they're actually aggregated and entrapped into what they call complex extracellular formations, namely beads and strings. And you can see in the pictures the addition
they have
of
lovely scanning EMs
beautiful.
Of these clumps.
Yeah, the bacteria are made into clumps. So that's what LCCL seems to be doing. And in fact, next
let me ask, isn't this sort of unusual? Michael, you would know. Can you think of other peptides or that do this to bacteria, aggregate bacteria?
No, the only
antibodies.
The only one that immediately comes to mind is the gram-negative Agarigati that facilitates aggregation amongst its friends, amongst itself. But I didn't know any single peptide that could cause microbes to almost aggregate, if you will, in a pure culture-like sense. It's sort of like quorum sensing.
Peptide.
It's sort of like quorum sensing, but not.
And it's a host factor.
And it's a host factor. It's not a homoserine lactone derivative. Remember, this is coming from cochlin, and it's a cleavage product as a direct consequence of the bacterial infection.
So this is pretty unique, isn't it?
Yeah.
In the sense that we discovered now, they uncovered a peptide that does this to bacteria, which is not the usual.
Well,
and
if you
at
think
the same
about
time, it's calling the neutrophils to the site where these bacteria
addition,
are now clumped.
It's a very smart peptide.
Well, thinking about it from an evolutionary perspective, hearing is our superpower. Without our ability to hear in 3 dimensions, we would be quickly deleted from a gene pool. So there is a strong selective pressure on the behalf of the host to preserve 3-dimensional hearing. And so it's selected for, and this unique situation of protecting this remarkable organ that, you have a tympanic device that is transducing sound. It's effectively transmitting waves that are being then picked up by hair cells that are responding to a wave front that then transmits the sound to our nerves. I mean, it's really quite remarkable of how it all stays together.
So, Elio, what came first, the timpani drum or the eardrum? I guess.
Well, who named the timpani?
Well, that's what I wonder if the.
Oh, I see.
If the drum was named first or the eardrum.
The drum was named first, I bet.
Yeah, because the drum's been around for a long time.
But the tympanic region looks just like a timpani drum head.
It does.
For those of you who have not seen it, it really does look like the drum head of a timpani drum.
That's very cool. All right, the last 2 experiments they do. First, they get an isolate of Pseudomonas from a patient who has had chronic otitis media for several decades.
Wow.
So there's a person who is either continuously or multiply infected and can't clear it for whatever reason. So they have an isolate of Pseudomonas from that patient. They show it can be aggregated by LCCL. So that makes me wonder right away, maybe this patient has a mutation in the gene encoding cochlin, right?
Well, and actually our first author, Ginseng Zheng, is an MD PhD who trained at Yonsei University College of Medicine, first his MD, and then he got a master's in oenterology, studying the ear, and then pharmacology. And he has a really nice publication record where he's trying to understand genetic hearing loss. So he's looking in patient populations, identifying mutations that affect hearing. And as you'll talk about toward the end of this paper, they're starting to think about how they might apply what they've learned here to treat certain hearing losses.
Of course, you could have other mutations or polymorphisms causing that as well. But since we've been talking about cochlin, that's an obvious one.
Right.
All right. So that isolate can be aggregated. And then they look at other bacteria that are involved in ear infections, Staph aureus, Haemophilus influenzae, Streptococcus pneumoniae. They're all aggregated by LCCL. So it looks like this could be a kind of broad-acting aggregator of bacteria that come into the ear.
Gram negatives and gram positives.
Right.
So
It's
a generalist.
It's effectively sweeping up the bacteria into a common pile to allow your immune system to come in and hoover them out.
Right. And they keep it in this scala tympani away from the
important bits.
The organ of Corti. Right. So that's the key here, which is beautiful that the LCCL goes only there. Why that is, I don't know if it's an anatomical thing or chemical or both, but that's really interesting.
Do you think those nearby hair cells are listening to the neutrophils as they chomp up and eat all of the clumps?
Maybe that's what the grinding is in your...
It's below the hearing threshold, Michele.
Probably.
The other thing I want to tell you, this is the last experiment. This aggregation seems to be calcium dependent. And why would that be important, Michael?
Again, calcium is important for a lot of gram-negative functions of the cell wall. That's the only thing I can come up with without looking more at what is going on with the intracellular milieu of all these proteins going on. But it may be divalent cation dependent. You could add, I suppose, EDTA to the experimental mix to see if
They
did. That's the experiment.
They did that. That's the next experiment.
Yeah, that's the experiment. Add EDTA and it inhibits the aggregation.
And they made that prediction by the primary sequence of this LCLL.
They also show that the LCCL directly attracts neutrophils by chemotaxis. It's a chemoattractant, which they can do in an assay, a cell culture assay. So that's essentially the story. There's a lot more to be done, but basically the LCCL is produced by proteolysis in the ear, the inner ear. What triggers the proteolysis must be the presence of bacteria initially, but we don't know exactly what. So that's something interesting to sort out. The LCCL goes into the scala tympani where it aggregates bacteria that attracts neutrophils and macrophages and other cells that are involved in its clearance, and it protects the auditory structures. So that is the question they wanted to address initially. And this is a mechanism, how it protects it by keeping, as Michael said, it sweeps the bacteria away into the scala tympani.
And allows the immune system to deal with it. And it also isolates the immune system from doing any scarification of the important bits that facilitate hearing.
That's right. That's right.
They also point out that the architecture of the ear also separates that compartment from our blood flow and the white blood cells that usually we think of as being recruited to a site of infection. So they need, both the eye and the ear need to have this self-sufficient innate immune response.
That has a name. It's called the blood labyrinth barrier.
Mm-hmm.
Yeah.
As opposed to blood-brain barrier, an equivalent of that. So there are other compartments in the body which are restricted and separate from the bloodstream, and this is one of them. I didn't know that. It makes sense, doesn't it?
Yeah, really thoughtful discussion. I learned a lot from their paper.
Yeah.
As did I. And one of the things I was scratching my head, since they were principally talking about Pseudomonas, but as they got into the next bits talking about Staphylococci and the Streptococci, that takes the outer membrane out of the equation as being contributory to this aggregation. And it begs the question, could it be something in the cell wall precursors that may be sloughing off as a part of cell division or something as cells divide? Maybe a couple of the disaccharides that are involved in extending the cell wall may be part of that triggering event.
Yeah.
So there's some evidence that there's been previous evidence that cochlin is involved in hearing in humans. There's a mutation in the cochlin gene that's associated with hearing loss, and they really don't understand how it works, because in mice, when you delete cochlin, the mice hear just as well as wild-type mice. So they're trying to sort that out and see what's going on. And they suspect maybe there's a dominant negative effect. Maybe the altered protein precipitates or aggregates and loses its function that way. But so this is going to lead them into more investigation in that area, I would guess. And it's quite interesting.
Yeah, they also did an experiment in mice where they first infected with Pseudomonas, and then they treated with this CCL and found that augmented clearance. And so they end their discussion talking about using this LCCL peptide as the starting point for therapy for people with chronic ear infections that lead to hearing loss.
I guess you could just put it in on a Q-tip, huh?
Yeah.
Peptide, because you wouldn't take it orally, I would think.
You'd probably just put in drops.
Put it in drops in your ear, yeah.
Yep.
Yeah.
That would be interesting, wouldn't it?
Really beautiful.
It's lovely. So I liked it because it's not just microbiology, but it's interfacing with the function of an important organ, in this case, for our hearing. And I think that's pretty neat. I think
ear infections are a big deal. They are a big deal, especially in the pediatric population, as any parent will attest.
As I said, I hear you.
And this paper also really pulls together anatomy, histology, immunology, cell biology. It's really, and microbiology, of course.
To think of this separate compartment in the body, which has its own limits, its own barriers. It's really, it's fun.
Yep. It's very cool.
Great choice, Vincent.
Yeah, thank you. All right. Let me read just a couple of emails here about recent episodes. So we have one from Natalie who writes, "Hello, Twimmers. In your recent episode about gingipanes and Alzheimer's disease," that was our last one, Michael pointed out that in the context of human health, neurological infections of HSV1, in addition to the episode focus of Porphyromonas family bacteria, can both be causes of AD. He suggested a possible future polytherapy of acyclovir and Core 388, which could be used to treat or prevent AD. I think it's an excellent suggestion, but I have a minor quibble. Oral acyclovir, while arguably effective, has the drawback of very poor bioavailability. Some indications require a patient take it 4 or 5 times a day. For most patients, this is a daunting, if not impossible, drug regimen, to say nothing of the challenges of use in the geriatric population. And there is the argument that any frequency of administration over 3 times a day is unethical for these and other reasons. Here's an interesting blog post by a physician trying and failing a QID antibiotic regimen 4 times a day.
Which is 4 times a day.
Yep. Even valcyclovir, a more realistic option, which is more orally bioavailable, by the way, would require patients to take it up to 3 times a day for full suppressive activity. Maybe at some point we'll have something better than that. Anyway, thank you for putting out such consistently fascinating podcast episodes. They are engaging in a way that frequently leaves me feeling hopeful about my own aspirant field, medicine. Keep up the good work.
Well,
Thanks, Natalie. You too.
that whole episode was reflecting around the bioavailability of the inhibitor that they had developed. And I did not know about the bioavailability of acyclovir or famciclovir, but any card-carrying pharmacologist out there would probably know those things. And I think if they're going to come up with a combination therapy, I think that is the subject for clinical trial in developing derivatives of acyclovir, famciclovir that have better bioavailability, because they well understand the mechanism of action of those drugs against herpes. It's against the Vincent, right? The thymidine kinase.
Yeah.
Of those, and so I think the chemists will just have to get busy and increase the bioavailability, as did the chemist at the company that developed Core 388.
So acyclovir and the derivatives are phosphorylated in infected cells only by the thymidine kinase gene. So they are not phosphorylated in uninfected cells.
Correct.
And in the infected cell, once they're phosphorylated, they are then phosphorylated 2 more times, and they're incorporated into the growing DNA change, and they cause termination. They're chain terminators, but they're very specific because it only works in infected cells. Now, acyclovir and valcyclovir work well enough to treat topical herpes infections, and they're even used to treat herpes encephalitis. So for that, they're good enough. Alzheimer's may be another issue, but Natalie, there is a clinical trial ongoing here at Columbia where they are giving early onset Alzheimer's patients, I think, 4 grams of valcyclovir a day for 2 years.
Wow.
And so, your point is well taken, but this is going forward anyway, so it's very interesting. Okay, we have Michael, why don't you take the next one because it has to do with floss.
Me? Okay, so Volker writes, Dear Microbials, thanks for your podcast. I'd like to ask Dr. Schmidt why he repeats that flossing is helpful. Wikipedia and a quick search at PubMed did not show me any evidence. Thanks and best regards. Well, candidly, the answer is my mother told me to. And my mother had to have periodontal surgery in her 40s, and she had 5 children, and she told her 3 sons that if they didn't floss, they would undergo the equivalent of childbirth 5 times in terms of pain associated with dental surgery to alleviate her periodontitis. And I, along with the American Dental Association, still advocate flossing, even though the clinical trials that have been done illustrating that flossing didn't provide any demonstrable benefits. They believe, though they haven't done the definitive studies, that flossing does have benefits. And when you talk to dentists, they anecdotally report that people who floss have less periodontal disease. Those clinical trials are challenging to do because people lie unless you're actually in the bathroom with them after they brush their teeth as to whether or not
Who
they
among
floss.
us has not fibbed when their dental hygienist has asked about flossing?
That's right.
Yes.
That's right.
Do you floss every day?
Every day.
And of course, we all swear on a stack of Bibles that we do indeed. But I think, candidly, looking at it from the perspective of just what floss does is it removes debris between the tooth enamel and the periodontal space, and you're pulling out the debris to retard the growth of plaque. And any periodontist will tell you what they principally do for a living is they scrape the biofilm off of our teeth and gum surfaces. And so floss is just
the hygienists do that.
Mm-hmm.
Well, the periodontist gets paid big bucks to do that.
I know, but I don't pay them. I pay the hygienist.
You pay the hygienist, but
that's the same thing. By the way, water pick is supposed to do some of the same thing as flossing.
It does, but you require an expensive device, and water picks are indeed good as well.
Not that bad.
So I put in the show notes the press release from the ADA specifically addressing the clinical trial that was completed in 2016 that effectively set the world on fire that flossing isn't important. So the flossers who lied to their hygienists were relieved, but the ADA still thinks flossing does more good than harm.
So let me ask you, Michael, someone told me recently not to use mouthwash because it ruins your oral microbiome. Is that true?
I don't know about that. I haven't seen those studies to know.
Okay.
But again, imagine that the mouthwashes either have alcohol as the antimicrobial, so you're going to attract beetles, or they have quaternary ammonia compounds in it, which are disinfectants. So you are indeed reducing the population. But I don't think the mouthwash is in there long enough, regardless of the antimicrobial agent, to it's sort of like mowing the grass. You're trimming it, but not completely debriding
Got it. All right. Michele, why don't you take that last one?
From Dawn?
Yeah.
Dear TWiM team, my name is Dawn. I'm an avid listener of the podcast and a self-proclaimed bacteriophile. Hopefully that makes you all chuckle.
It did.
I truly enjoy learning about microbiology. I only wish that I had realized my passion for microbes at minimum a decade sooner. After earning my graduate degree in curriculum development and working as an educator in teaching English to non-native speakers for almost 10 years, I'm now a student again. I endeavor to become a nurse and eventually a nurse practitioner with a focus in gerontology. In addition to being a student, I coach women's competitive gymnastics as well as instruct adaptive gymnastics classes for children. My class is full of amazing individuals ranging from 7 to 15 years of age. The majority of the class has autism, ranging from nonverbal to high functioning. In the future, I'm keenly interested in doing research. I'm particularly interested in both autism spectrum disorder and neurodegeneration and the role that microbes may play in both of these processes. From the very moment I hear your voices, my heart swells with joy. I feel as if I'm hearing from an old friend. I am so grateful for all that you do, your enthusiasm, tenacity, work, and productions. I just want to say thank you. Here are a few photos, so this ain't just another boring email. I hope you enjoy. My Unknowns project is one of my greatest accomplishments, as is the dichotomous key which I created. I have a profoundly inspiring but rigorous professor for my microclass, and I can honestly say that I literally put blood, sweat, and tears into that experience. Please keep what you're doing. You truly make a difference in the lives of so many people. Respectfully, Dawn. And then she shares some photos. Would anyone else like to describe these?
So one is her paper on this unknown, which she took for her class, 79 out of 80. Great work. So she's very proud of that. And then
she had Citrobacter frundii and Staphylococcus epidermidis.
That's right. And then she showed a picture of some of her streakings, I suppose, there.
That's it.
Right. Yeah.
Yep.
She sent
isolation for single colonies.
Yeah. It's cool. Yeah. That's great. Well, thank you.
Great work.
Dawn. Great work. Keep it up. Glad we inspire people, right?
Oh, boy. Thanks a lot. It helps to hear this, doesn't it?
Yeah, it's great.
She's obviously a talented and disciplined individual, so I'm sure you've got a bright future ahead of you wherever you decide to go.
Just keep listening, Dawn. All right, that's TWiM 196. You probably listen on a phone or a tablet. If you do and you use a podcast player, likely, please subscribe. We'd like you to subscribe. You get every episode as we release them, and we know how many people are listening. That helps us. If you really like what we do, consider financially supporting us. You can go over to microbe.tv/contribute, and we have a couple of ways where you could, say, give $1 a month at Patreon or PayPal to help us. And, of course, we love getting your questions and comments. TWiM at microbe.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
Go Blue. It's game day. We're in the
It
Sweet
is
16.
indeed. So any predictions?
Blue all the way.
Blue all the way.
Elio Schaechter is at Small Things Considered. Lovely blog. Thank you, Elio.
Thank you. So appreciate it.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.